Good morning, good evening, good afternoon, everyone. Welcome to Ecology Live. I'm Mark Cadot. I'm Chair of Applied Ecology Resources, a new online resource, and I'm also the Editor-in-Chief of Ecological Solutions and Evidence, a journal published by the British Ecological Society. If this is your first time joining us, you're very welcome. And welcome back to all the Ecology Live regulars. Also, thanks again to NHBS, who are generously sponsoring today's lecture. They supply equipment and books for ecologists and conservation professionals. They're offering free European shipping for Ecology Live viewers and have extended this offer until the end of September. There'll be details at the end of this talk about how you can take advantage of this. If you're enjoying the Ecology Live talks, make sure you check out what else the British Ecological Society is offering. For example, the Teaching and Learning Special Interest Group is holding an online webinar to discuss the creative ways fieldwork teaching activities can be delivered during COVID-19. And our Aquatic Ecology Special Interest Group is holding its annual conference at the beginning of September with some amazing talks, workshops, and a Twitter seminar where early career researchers will present their work. If you're interested in these events, make sure to register before the deadline on Friday. All the details of this and other events are at www.britishecologicalsociety.org/events. Make sure you check it out. I'll shortly hand over to our speaker, Maydianne Andrade, who's giving today's seminar. But let me first quickly say that we are recording the talk and will post the video to YouTube afterwards. Also, there will be a short question-answer session at the end of Maydianne's 25-minute talk for those watching live. Please do submit your questions during the talk using the Q&A box. There's no need to wait to the end of the talk. These questions can be named or anonymous. You can select. We'll then select a couple of questions to ask our speaker at the end of the session, and we'll see how many we can get through. So without waiting any longer, I'm very happy to introduce today's speaker, my colleague and my friend, Maydianne Andrade, who will be going to explain how widow spiders can help us explore the evolution of plasticity. Maydianne, over to you.
Thank you, Mark, and welcome everybody to the corner of my dining room in Toronto on the east side of the city. And for me here, it is morning. So today I want to talk to you about work that we've been doing in my lab and my group over the last really couple of decades on widow spiders, or black widow spiders as some people know them, and why we think they are good models for looking at plasticity. Now, I'm interested in behavior and understanding behavior in its ecological context. And typically, behavior has been thought of as a way in which animals can interact with that environment using the tools that they have in terms of their physiology and morphology, which have been developed with a particular life history. And the idea then is that there's sort of this flow from what you have as a tool to behavior, which can then adjust your fitness outcomes in a given environment, where fitness might be thought of as a number of surviving or lifetime reproductive success of you and your offspring. Now, what's clear and has been for some time is that it's not as simple as that, and part of the reason for that is that the behaviors that are necessary to accrue fitness advantages in a particular context have a particular shape, but may not be consistent across contexts or across environmental conditions. And so here you see, for example, a male whose fitness is at the lower end of the distribution relative to competitors in a context that may involve the presence of a large number of females. And my focus is on mating, so I'm thinking primarily about how males acquire reproductive success. But if you change the context so that now the grayed out females are not available for mating, so there are fewer of them, and there are more males, perhaps this same male with the same set of morphological and physiological traits, the same life history distribution, will end up at this point of the fitness distribution. That is, this individual will now have higher lifetime reproductive success than its competitors. And recognizing that effect means that instead of it just being a flow into plastic behavior or variation in behavior to fit an organism to its environment, there may be evolutionary effects where if there is a predictable match between the traits that are necessary to acquire behaviorally fitness, then there may be an evolutionary feedback that actually adjusts things so that during development, an organism may develop different sets of traits depending on the environment in which they are developing. And that is plasticity, developmental plasticity, or adaptive plasticity. So same genes, different traits resulting. So I've become very interested in this because of the need to try to understand how this works in nature, which organisms will be plastic and which won't, will have implications for our understanding of which organisms, for example, will do well despite environmental change and which will be unable to be resilient to those changes. And so what we expect if there's adaptive plasticity is that it would occur if there is information available on the lower left here, developmental inputs that reliably predict adult environments. And adult environments are variable so that they're not predictable, for example, by the environment of the parent. But if there's developmental inputs that individuals can detect that will predict the adult environment, then we would expect over evolutionary time the developing juvenile in some species will evolve to detect those and will alter its development and its adult phenotype so that what you see as a result is that the adult phenotypes that result after detecting information and changing development will have a fitness advantage relative to organisms that do not engage in that sort of plastic cycle. And so what we have is ecology and evolution coming together to result in adaptive plasticity. But all pieces of this cycle are necessary to demonstrate that plasticity exists and that it's adaptive, and we need to understand when we expect this cycle to happen and to have this ecological evolutionary linkage. One classic example of adaptive plasticity is in Daphnia. So this is an example from a paper in Science from many years ago. On the left and the right are clones. So these are genetically identical Daphnia. These are water fleas, essentially. The one on the left was developed in the presence of chemical cues of a predator. The one on the right did not have such cues. And you can see clearly the adaptations that have developed in this organism, despite having the same genes as this one. And in fact, this one has lower susceptibility to predation as a result. But these are energetically costly. So there's a trade-off for building these traits. And it makes sense to, or at least in evolutionary time, you may see a link in which these develop only in the context in which they are needed. Otherwise, those resources will be shunted to other things. So my interest in plasticity, as I said, is in mating behavior and how that may drive plasticity in behavior, physiology, and morphology. And today I'll be talking mostly about males. And what males are doing depends on what other males are doing in terms of what will yield the highest fitness, but also what females are doing. And the context I'm interested in that I'll talk about today is essentially demography. I'm going to use the terms about the environment quite loosely. You might be talking about population density, the proximity of conspecifics who might be competitors or potential mates. But the basic idea is high density, close proximity to conspecifics, or low density and conspecifics kind of far away. So if you're talking about understanding male reproductive traits and want to think about predictions about plasticity, what I need to be able to do is make predictions about the different contexts in which males might find themselves. And then what do I expect in terms of shifts in their life history and the resulting adult traits. Now that requires thinking about how males divide up the resources they have to produce reproductive traits as adults, to develop and grow, and to maintain their soma. And making these predictions can be complicated because, of course, this pool of resources for most organisms is constantly replenished through foraging. And many males engage in repeated or multiple matings over their lifetime, and those matings may occur in different contexts as males move through their environment and as they age. So this is a complicated thing to try to make these predictions. And that's why I think that black widow spiders, the widow spiders, are so useful here. This is a group of organisms in which many of them engage in terminal investment. That is, rather than mating repeatedly with variation in context across those matings, males mate only once, typically in their lifetime. Right away, that constrains the context in which males have to perform well to gain reproductive success, and we can then make evolutionary predictions about what we expect in a given ecological context. So there's a variety of species in which males are single mating specialists. They are It is not uncommon in spiders among relative to other taxa. So here you see a number of them, including on the lower left Latrodectus, which are the widow spiders that I study. There's a variety of other great studies on different types of terminal investment strategies. With Latrodectus, and especially the Australian redback spider and the Latrodectus geometricus, which is the brown widow spider, what is very notable is that the male actually has a posture during mating that makes it easy for the female to consume him during copulation. And so males typically mate only once. I won't go into details about these others, but I encourage you to look them up. So the types of traits that we see in species with terminal investment show some commonalities. Some of them have behaviors that are clearly make it easier for the female to kill them. Some of them die spontaneously, even if the female doesn't kill them. That is, they clearly have a life history that's tuned to a single mating. Many of them have genital damage or mutilation that may make it hard for them to mate again. They show permanent sperm depletion, where there's no sperm left for subsequent matings. And they also show extreme sexual size dimorphism. I don't have time to get into all of these, but some of the key pieces here for our predictions is that in addition to mating only once, males of these species tend to have very short lifespans. So again, that restricts the context in which they have to compete. And they're capital breeders, which means they don't eat after adulthood. So there is no replenishment of nutrients. So we can make predictions based on understanding in a fixed way how much they have to allocate to these different life history traits. So the advantage of working on terminal investment species like the widow spiders then are a simplified adult context, which simplifies predictions. And also, because they are short-lived, it's possible to have reliable cues of the adult context available to juveniles because they don't live for a long time. And so what they detect as a juvenile may tell them reliably about what's going to happen to them as adults. And again, I'm using verbal shorthand, but I mean over evolutionary time, this would favor males who are able to detect something in their juvenile stage that then is relevant to the competitive context as an adult. So let me tell you about black widows in general, but also redback spiders and western black widows, which are the two that I'll talk about in detail today. And then I'll talk about sort of a case study of understanding variation in male size using theory around plasticity. So the black widow spiders are a group of 30 species that are distributed worldwide, as you see here. My students and I have been studying them in various places in the world for many years. This is me as the cartoon, but my other students, my students and postdocs are here and they're fantastic. Now, the interesting thing about widow spiders is that they all produce their egg sacs, as you see here. Females can produce up to 300 eggs per sac, and the individuals in those sacs disperse by ballooning. Now, ballooning occurs when a little bit of wind carries a spider away. And what's important about this, and this is from a study a couple of years ago, what's important about this for our purposes is that it means that the environment in which a male finds himself is unpredictable. The same is true for females. So they disperse randomly through the environment, and there's no way in which the adult context predicts the context in which the juvenile will be competing. Now add to that when males become sexually mature, they leave the web in which they develop and wander off looking for a female, and they have extremely high mate-searching mortality during that time. At least in these two species and a few others where it's been measured, it's around 80% or higher. For redback spiders on the left, there's also sexual cannibalism, which I alluded to. At about 86% of males are killed during mating, so males really only mate once. For western black widows on the right here, they also have high mate-searching mortality, but they are able to mate more than once if they manage to make it to another female. They also have a slightly longer lifespan. But as you can see here, although females can live up to 2 years, at least in the lab, males live for only about 25 days for redback spiders and about 90 days for western widows. So again, constriction in the context in which they'll compete. Their mating behavior, I need to talk about this a little bit because the other piece we need for predicting plasticity is understanding what they need to do well, that is to acquire matings, and what would there be selection over time. So one of the things about widow spiders that's interesting is that they have prolonged costly courtship. So they court on the web when they reach a female, and they'll do that for half an hour to actually 2 hours before they even contact the female. And then they spend more time on the female's body engaged in courtship. There's this extreme dimorphism, so it may be hours before they mate. And we know that's costly because they're producing vibrational signals throughout this time. And so in this great video from Sen Sivalingam, laser vibrometry is used so that you can see and hear the male's courtship. It involves vibratory signals. Sen is just writing up his PhD now, and he has shown that these signals are stereotyped and produce information about male body size. When the male gets on the female, there is sometimes other males present, so it's not just a matter of making it through this courtship, but males may have to compete, as you see in this video by Sean McCann. One male is beginning to mate with a female. This is the male on the left. This male on the right is competing with that male and will actually bite him, and you'll see that the male who was beginning to mate will die. So this competition is actually quite stiff. And that means that there is a variety of things that males need to do, both that would allow them to court, that would allow them to compete with other males if they're on the web, that would allow them to convince the female to mate with them, and all that after they've had a costly and risky trip to find the female in the first place. So when they mate, spiders mate actually with structures at the front of their heads, essentially, or cephalothorax, and there's two of them. These are called pedipalps, and they correspond to two sperm storage organs in females, these dumbbell-shaped structures on the right. They're accessed from the underside of the female's abdomen, from the ventral surface, and males inseminate each of these independently with each of those copulatory organs. We know that mating with a female first is important because males actually leave behind the tip of their copulatory organ. On the left here, you can see that there's a difference between a male who's unmated on one side and is mated with this side, and the very tip where a sclerite has broken off and remains lodged inside the female's sperm storage organs, which you see on the right. This is from work with Lindsay Snow and Emily McLeod. The result is that males have to engage in scramble competition. What does that mean? When a female becomes sexually mature and is unmated, the male who gets there first and manages to mate with that female is going to have a big advantage in terms of paternity. About 70% of the time, he will successfully place plugs that prevent other males from inseminating that female. This is the family tree of the black widows. And so just to show you why this is perhaps a good taxon for working on these questions, there's variation in the occurrence of monogyny, or the case where the male mates only once because of these adaptations I showed you for terminal investment, and also cases where males are polygynous, where males are able to mate with more than one female, even though some of them may have high mate search mortality. And so this just gives you in a truncated tree a feel for how that's distributed, and that it has this geographic, it's not all geographic variation, even sister taxa can show differences in mating system. And this is a truncated tree produced by Charmaine Condi. She's working on her PhD right now. We'll soon have a more up-to-date tree to share with us. So in the lab, we do a variety of studies. We raise species in the lab at high density. I like to say that I have the largest captive population of black widows in the world. Maybe someone here has more, but when I'm at full speed, not under COVID, it can be upwards of 50,000 spiders or more. And this allows us to do a wide range of different types of studies. And here you see some of our macro videography by Sean McCann. We do a lot of work with pheromones and signaling to try to understand the cues that males may be able to use to detect things about the environment that are relevant to reproductive success. This work is done by Luciana Barafaldi and Catherine Scott, among others, and we are able to extract pheromones, and together with a collaborator in Germany, Stefan Schultz, we've characterized some of the pheromones for some of those species. So our work with pheromones shows us that there are cues that males could use to detect the density or proximity of potentially unmated females, again, those females that the male could mate with and get high paternity with. And those are available to males during development. And that's because females, when they're unmated, produce airborne pheromones that do attract males from a distance. But once that female mates, the pheromone production stops. So females that are mated and potentially plugged are essentially invisible to males. So males have this opportunity then, when females become sexually mature, to try to be the first one there. And again, try is colloquial, but evolutionary adaptations that would allow them to engage in scramble competition to get to that female first. So that gives you a picture for some of the things that males are dealing with. Scramble competition, but if there's another male there, there's also direct competition as well. So let's look at male body size and think through how these things may manifest in terms of plasticity. Now, the reason we were interested in plasticity with widow spiders is because, indeed, there is a lot of variation in male body size. Here you see two male adult black widows on a web with a female. These are Western widows. And we've got about a 45 milligram relative to a 9 milligram male, and that is not at all uncommon in the field. So wide variation in body size. And in fact, if you look across species, and this distribution is actually for 210 different species across different taxa, from mammals to fish, what you see is that Latrodectus species are on the upper end of this distribution of variation in male body size or mass. And the champion here is actually the northern widow, which lives here in Canada and some places in northern US. So very high variation in male body size is common. How do you understand where that comes from? We don't expect to see the maintenance of such high levels of variation necessarily. And so with Michael Kasumovic, a former student who's now professor in Australia, we looked at the possibility that what we were seeing was plasticity being driven by trade-offs between development time and male adult traits, body condition and size primarily. Because for invertebrates at a given level of resource availability, if you develop quickly, then you have more time to acquire or you have less time to acquire resources, and your size and body condition are lower as a result when you become sexually mature. If you develop on a slower time scale, then you can be larger and be in higher body condition. And so what we predicted then is that males may be detecting their local environment and whether scramble competition is important, or whether in fact they were moving into a low density situation in which they would have to endure long distance mate searching and perhaps compete for females. So at high densities, we expected development time to be favored, rapid development time, and the result would be small size and body condition. And if there's variation in this across seasons or across habitats, then we might expect to see the maintenance of those different body sizes in nature. So males are doing different things. So just to remind you then, for development time, because we know that sperm competition favors males who arrive first, if females are becoming sexually mature at a high density nearby, males might accelerate their development and the cost would be small body size. But there is a trade-off here, because we know that in terms of body size, larger males do better both in terms of female competition and choice. Male competition and choice. So I'm not going to go through these data in detail. They're quite old, but I've got the references here if you're interested. So in terms of endurance and direct competition, a series of studies shows that in general, large males will win over small males. So indeed, there's this trade-off between development time and size. I won't get into body condition, but it's very similar. So increased development time would mean increased size, but again, that wouldn't work if it's scramble competition in terms of increased fitness. So what are they using to detect female density? As I said, we predicted for redback spiders at least that it's pheromones, that they are actually able to detect the proximity of females who are recently matured, and that would be the developmental input that leads to developmental plasticity. In other species, while those pheromonal cues are still present, if they are highly seasonal, it may be that seasonal variation in female density is actually more important, and that male development will be entrained to abiotic cues that indicate phenology, or where you expect the highest proportion of immature or recently matured females to be arising in the population. So we've done field work on Vancouver Island in BC. Catherine Scott and Sean McCann did this work over the long field seasons. And I want to thank here the Sawat First Nation for allowing us to use their lands. This allowed us to track what's going on in the field. And the data from those studies shows that, in fact, there is a regular seasonal pattern of female availability. So in this population, for example, receptive females are shown on the bottom, and males who would be competitors above. And you would predict early in the season, males need to have endurance or direct competition phenotypes because there's very few females for them to find. Later in the season, in the fall, high densities of females would favor scramble competition. You need to get there first before your competitors. We've also done field work at the Hastings Natural History Reserve in the Carmel Valley. Sheena Fry, Maria Madanu, Emily McLeod, and my current student Nishant Singh have worked there. And we have a multi-year data set that shows that we see something similar in that population of black widows as well. So in the fall, most females are unmated. That is, they are available for mating, and these males may be the first one to mate, and thus have highest paternity if they manage to get there first. So scramble competition be favored in this period. There's then a period of inactivity in the winter that kills a lot of males and sort of resets things. And then the new cohort of males, if they are present in the spring and summer, what they will encounter is a very low proportion of unmated females. And so in this context, we expect males to need endurance phenotypes. So how do we test these predictions? We can do it in the lab and the field. And again, this piece is older work, so I'll just summarize it. And what we did with Michael Koncibovic and Jeff Stoltz, we've replicated this several times now, is expose males to the pheromones of females. And this was in an environmental chambers in which the males were not able to contact the females but could smell their pheromones. Or we expose males only to other males. And what we did was find that consistent with our predictions, when males are not exposed to females, they develop more slowly, they end up larger, and they have increased longevity as a result, compared to when there's females present when they did engage in rapid development and were smaller. For Western widows, more recent work, we examined similar kinds of questions, whether there was in fact developmental links that led to high-density scramble phenotypes versus low-density endurance phenotypes. And for this, we, instead of rearing them in the presence or absence of females' pheromones, we reared them in the presence or absence of the abiotic cues that we know indicate the time of the season. And so both temperature and light cycle were manipulated, and we quantified a number of different phenotypes in the males as a result. And what we found was that, consistent with our prediction, development time in the fall was more rapid, and that is the period when there are a large number of immature or recently matured females who would be available to males for high paternity. And in fact, as a result, they grow less, they have smaller body size in the fall compared to the spring. And we did this on 2 different diets. I don't have time to talk about that today. So this is consistent with our predictions. We also looked at how this played out in the field. So doing this in the lab is one thing, but really the field data tells us whether this is happening in the field and how it affects fitness. So in the field, if you collect males in the fall and the spring, the fall in green and the spring in yellow, and this was done over 2 different years, you do find consistently that males that are collected in the spring are larger and actually in higher body condition than the males that are collected in the fall. Again, consistent with our predictions that fall males are adapted for scramble competition, rapid development, small body size. We also asked whether there's evidence for fitness effects of this. And so what we looked at was in field experiments where we placed males out in the field, we placed female webs in the field, and female silk produces pheromones that are released, as I said, that attracts males. So by doing this, we put out the webs of recently matured females who are releasing pheromone. That simulates a female who's just become mature. So there'd be scramble competition for males who are naturally occurring in the field for that female. And what we monitored was the order of arrival of males. And based on order of arrival and a variety of laboratory studies, we were able to predict what those males' paternity would have been if they had been arrived at the female's web in that order. So these are the data from those studies done over 2 years. And what we found was on the x-axis, male body size, this is a log scale, smaller males on the left, larger males on the right. And the proxy estimates of fitness from the 0 to high fitness shows that, consistent with our predictions, despite the fact that large males we know have advantages in direct competition, small males are the ones who have the advantage when it comes to scrambling for a newly matured female. So we've then looked at all pieces of this cycle in 2 different species in widow spiders, in western widows and in redbacks. And although there's a lot of work to be done, we feel that we now have a good feel for the variation in adult environment that affects male fitness. And this is related to the density of unmated females or recently matured females. We know that there are developmental inputs that males pay attention to, that is, in terms of shifts in their development. And those are both seasonal and sex pheromones. We know that they're detecting this information because we've done experimental studies of this, as well as seeing that's consistent with the field results, and that this does result in changes in their adult phenotype as well as their development time. And we've seen evidence that there is a fitness advantage for making this shift. There's a lot to be done still, but understanding this has really taken us to a place where we can begin to ask some really interesting questions about the evolution of plasticity. We're interested in this for a variety of reasons. Eventually, I hope to be able to look at this in a comparative way across the entire clade and across the geographically variable habitats in which they find themselves. Plasticity, we think, may be related to divergence and invasiveness, and those are some of the questions that some people in the lab are asking right now. If you want to learn more about plasticity, I did talk about this fairly quickly. I do have a recent book chapter, which is a bit of a slog to read because it's really long, but I did provide that to the organizers of Ecology Live. And I want to end by thanking the more than 200 undergrads who over the last 20 years have been instrumental in raising those thousands and thousands of spiders that were necessary for their work, also in engaging in some of the research that was foundational to what we saw here. And of course, to my current lab and my recently graduated students, those who provide photographs and my funding sources, and my kids who were very nice about eating breakfast before this, so you didn't hear plates chinking all the way through. And that's it. And so I'd be very happy to answer questions.
Thank you so much, Maydianne. That was wonderful. And I'm sure everyone appreciated the wonderful photographs and the videos that people from your lab over the years have supplied you. That's fantastic. So there are some questions, and I'm going to ask them on behalf of the listeners, or the viewers. So one question by Edgar Gonzalez. He says, "Excellent talk, Maydianne." And he's wondering if you've done any population modeling to demonstrate the maintenance of high male size variability over time, and are there any other factors over long time spans or spatial scales that might maintain male variability?
Thanks for that question, Edgar. I have not done any of that modeling yet, and I would be very interested in a collaborator who would be interested in doing that. There are other factors that are important. So, for example, I alluded to the fact that they are multivoltine, and so, in fact, some males in aseasonal populations overwinter as juveniles. Some of them are from egg sacs that have hatched at the beginning of the season, but then once the season starts, egg sacs are hatching continuously. So it's actually a fairly complicated problem to think about how these different factors with spatial and temporal variability would lead to what we see, which is sustained variation in male body size.
That's actually a nice segue into another question, anonymous question. And they're asking, what happens if you, quote unquote, confuse them in experimental conditions by raising them in different environmental conditions, so autumn conditions versus spring, with the various parameters in your experiment, and they want to know, do different cues dominate under different environments?
We don't know that yet. So we are kind of confusing them by putting them into these environmental conditions, because we take males that have been reared in the lab so that we hope that most of the maternal effects are gone, and then we put them into these conditions. But then when they compete, they're under common garden environments. So we aren't giving them the advantage of competing in the context in which they actually developed. And that's one of the things we're interested in, but it's a very challenging logistical problem to crack. So hopefully stay tuned for that. We do have the ability now to change the temperature, for example, in our environmental rooms. And we know from work by Monica Mowry, who's now in Yale Lubin's lab in Israel, that temperature effects do shift development as well, and they do so independently of photoperiod.
Maybe one more question, just because given the time. In natural conditions, males are obviously going to be searching for females. What's the role of environmental heterogeneity or spatial heterogeneity, like different plant types, plant height, other types of small scale variability? Can that affect the maintenance of plasticity?
That's another great question. Everything's a great question. That's the problem here. So I'll just give you a little segue into this by saying that when I started working on black widows, maybe 24 years ago now, I would go to a talk and just say, I don't know, because we didn't really know very much other than some foundational work by Yale and a few others. Now we know a lot, but there's a lot we don't know. And in fact, one of the reasons I was pleased to get this invitation is that I need more ecologists. I have one ecologist in my lab now who's looking at some of these questions. We have a giant data set from 6 years of field surveys, but we don't know yet. One of the things that's striking, though, is that the male mate search mortality has now been measured in 4 different species in radically different environments, on a beach, in that grassy field you saw, in Israel in a desert, and the mortality rates are all around 80%. So we don't know what's going on. It's really interesting to think about.
Great. I mean, it's a fascinating system. Thanks again, Maydianne, for your time and a wonderful presentation. I can see other comments popping up in the Q&A saying, wonderful talk, thank you. So a lot of people appreciate this. And with that, I'll turn on my slides. Okay, so yeah, so thanks again, Maydianne, for a fantastic talk. Another great Ecology Live is coming to a close. Um, Next week at the same time, same place, we'll have Alexandra Harmon Threat, who will be giving us a talk about how conserving bees is a dirty business, which is very timely and also very interesting. So please don't miss it. Uh, and finally, I'll leave you with the details of the offer from our sponsor of today's Ecology Live talk, NHBS, so that you can take advantage here. And with that, everyone, have a wonderful weekend and see you again next Thursday.